And there were some pretty intelligent people doing this task. Believe me, there were some of our colleagues. And the capacity for these things is only about sort of 2.6 square, so it's pretty low. We did this task at the same time as recording MEG. So you probably all have seen EEG, because that's a very traditional, very powerful method where you put electrodes on the scalp and you're measuring the electrical activity produced during a specific cognitive task. Another technique, which say Anko has been working with for a long time, is MEG. So instead of electroencephalography, it's magnetoencephalography. So if you remember from being at school in your physics, so you have these right-hand rules. So any moving electrical current produces a magnetic field, and that's what we measure with MEG. Now, of course, it's a very small magnetic field, so we need a very expensive machine to measure it. And this is an illustration here of a MEG scanner. So you need superconductors. In fact, the technology was developed for airplanes to be able to see submarines. So they're very sensitive. They're called squids, or superconducting elements. So when you look at this is called a power spectrum. So this is looking at all the subjects and all the channels. And these are the different frequencies. And you can see here that during the task, during this retention period, there is a blip here at about 10 hertz. So that seems to be the dominant frequency that the subjects were using during the task. And we went on to look at individual people. So we split them into people that did very well and to people that did less so well, and calculated their bandwidth. And you can see that these subjects did better, had a bandwidth that's twice as big as these subjects who did worse in the task. And that was localized to the right frontal area. And you can see, again, right frontal and parietal. So the areas that are classically used during working memory tasks, we can use MEG to localize their activity in the frequency domain. And you can see here that the capacity of a given individual correlated with the power they had at this 10 to 12 hertz window. Okay, the last thing I'm going to talk about is a technique called transcranial magnetic stimulation. So, as I mentioned before, a moving electrical current in the brain can produce a magnetic field. And what we can use the inverse of that, and we can have a moving magnetic field to produce an electrical field. So what what TMS, or transcranial magnetic stimulation, is that you put a big magnet on top of someone's head, as you can see here. So basically, that is the big magnet tend to be in a figure of 8. So the figure of 8 serves to make the magnetic field very focal. So you have this magnetic field over the brain, and you generate this field on and off very quickly, and it produces an electric field below the skull. So it directly stimulates the brain. And it's amazing when you have it done to you for the first time, because you can have it over your hand area, and you can stimulate it, and you can see your hand move. So it's like a physiology in practice. Now, it tends to be used to study cortical function. So the idea that it can affect something deep down in the brain, like the hippocampus and the medial temporal lobe, seemed unlikely until there was this one paper published by Hartwig Siebner, who showed that when you do a PET study at the same time as doing this inhibitory type of TMS, so it's 1 hertz, so it slows the brain down, you also had a decrease in regional cerebral blood flow in the medial temporal lobe, in the hippocampus, which I thought was really cool. So we then did the same protocol of TMS, but we mixed it with a memory task that was just ongoing. So they were seeing lists of words and then recalling the words, and this went on during a baseline period, and then they either had real or pretend TMS during 12 lists, and then 12 lists afterwards, and came back for a second session. And what we found was that, okay, so this is the pretend TMS. So there's very little change from baseline in the effective TMS. But the real TMS had quite a profound effect on memory function, up to a 40% drop towards the end of the actual active TMS. So this sort of discovery is what we're intending on doing here in Madrid, is to use this to see if we can manipulate emotional memory, to see if we can decrease the strength of emotional memories. As you know, conditions like post-traumatic stress disorder, you have excessive memory. So this may be a potential treatment. So in general, the conclusions, as you will have seen, there are lots of possibilities to study the human memory system using functional imaging, a combination of functional imaging and other techniques. And those are just my collaborators. So you see although I'm Polytechnico, I'm working with quite a lot of people from the Complutense as well. Thank you.
Okay, thank you very much, Brian. So we have time for questions now.
Remove them. So at the moment the most promising ideas about treating post-traumatic stress disorder is this phenomenon called reconsolidation. So the classical view has been that when you learn something, that the process of memory, basically 3 stages. You encode, you then consolidate it, the memory, and then you can retrieve it. What was found probably about 10 years ago is that when you retrieve a memory, you make it fragile again, so you can affect memory, even though you thought that it was there really strongly. Just by bringing it back, you can affect it in a similar way that you could affect it when it was first encoded. So what we're looking at quite a lot here, and also in the Netherlands, and work we're doing in the Netherlands, is ways to harness this phenomenon of reconsolidation to eliminate specific memories. So the idea with post-traumatic stress would be to have them reactivate their memories, but then to apply a manipulation which could be a pharmacological manipulation, such as a beta blocker, because the beta blockers are in clinical trials now for post-traumatic stress, or something like TMS. So the idea of memory erasure is growing. I mean, it will probably be possible quite soon, because there's already quite a lot of work. You can certainly erase memories in rats. Well, I guess it's difficult to prove that something's not there. You know, the statistical inference of erasure is not easy.
Okay, more questions? So I'll ask you, Brian, about the part where you talked about whether you were going to remember something that had been shown, and you were showing that there was activity in the perirhinal cortex when this happened. I was thinking of work where they show that when something is remembered, there is more synchronization between neurons in the hippocampus than for items that are not remembered. I wonder if there is a relationship between the two effects.
Yeah. No, it's a good question. So the work I showed you by the German group was published in 1999 in Science, and 2 years later they had a Nature Neuroscience paper showing synchrony in the theta frequency between the perirhinal area and the hippocampus during successful memory formation. So absolutely. Now, I don't know what synchrony means. Yeah, I don't think anyone knows what it means. So what we're talking about synchronicity, if you have, I told you that all the brain is, different parts of the brain is doing this all the time. Their electrical potential, their activity is doing this. And synchrony would be that they're doing this at the same time as opposed to being out of phase, so they're somehow coupled, although they're in different places. The activity resembles, the waves are resembling different parts of the brain. But there's a lot of interest in synchrony, but I can't tell you what it means. They're doing the same thing, but we don't know what that is going to mean in terms of bringing the two parts of the brain together for the future.
More questions? No? Okay. So thank you very much. Bye.